Hi, everyone back at home, please do not put your kettles on quite yet because we've got an amazing group of contributors in the studio right now. Now, over in the live lounge today, we are commemorating 10 years of London Calling by sequencing live the genomes of 10 critically endangered species. One of those is the rockhopper penguin, where the sample was donated very kindly by Edinburgh Zoo. And that really draws us into the theme of one of the talks that we've just had. So I just want to welcome to the sofa today. So we've got Carol Grier and Daniel, both from the University of California, Santa Cruz, and Mrinalini Watsa, who's come all the way from San Diego Zoo Wildlife Alliance. Welcome to all of you. Just keeping on the theme of conservation, my first question is going to be for Mrinalini. So one of the most inspiring parts of your talk was when you were talking about slowing down to stay local. And I think we've heard in all of your talks this morning that accessibility is really part of your scientific thought process. So we really strive hard to make technology that is accessible, that we can ship anywhere, that is accessible in terms of price and also utility. What does it mean for conservation to have accessibility and have that local sequencing, but also the local bioinformatics by the local communities?
I think that for ages and ages, really, when any kind of biological exploration, has taken place, it's been done in a certain way. And when I was a graduate student, I would hear about this all the time, right? You go to a country, you collect, then you spend hours and hours trying to figure out how to get the sample back to your lab. And every time you do that, no matter how many people from that community you involve in your research team, they could never make it back. And so in the early days, I tried to fix that. I said, well, come to my lab in the US. Maybe that's a good way of doing it. But we couldn't get visas for people to do that, right? And so I think we just got stuck, and it just became like the inevitable thing that no matter what you did, you were always taking samples outside countries and outside communities to these labs far away. And it made it such that anyone local could only participate in a certain part of the pipeline, and then their involvement ended, right? And then they never got to the point where they could do anything exciting. And they started, I think, almost to believe that maybe they couldn't. That this was just some complicated, highly scientific, very technical thing to do, and it's really not. It's very, very doable. If you can cook, you can do genomics. It's not really more complicated than that. It's a recipe and you're following instructions and it can be learned. And so I think when Nanopore came up with these movable devices, when they were $1,000 to buy, right? It just changed everything for us. It was suddenly said, that's it, that's the last obstacle. Now we have absolutely no more excuses. We have to do this locally.
Fantastic. And Carol, you shared with us your incredible Nobel Prize-winning science in telomere science. And I think everyone in this room is talking about telomeres at the moment. And one of the most interesting things about your recent paper for me was the fact that you did it on a MinION. And so what does that mean in terms of how that research could be brought forward into affecting people's lives?
Yeah, we are really excited to be able to make this kind of analysis available to many different laboratories, that it's really low entry to be able to do this kind of telomere analysis, because the analysis up until now, in terms of the clinical analysis, has been just done in a few labs. And in terms of the basic science, there really isn't that nucleotide level resolution for measuring telomeres. And so we're really hoping that this will be accessible to many different laboratories. They don't have to have a Promethean or anything like that. They can just low investment get the MinION. And so we're hoping that many different people will adopt this telomere sequencing in labs all over the world.
And there were questions from the audience about degeneration and how you might be able to understand the risk to particular patients, particular people going through their lives around what information telomeres give to people. What are the kind of assays 10 years from now that you could imagine people using telomeres to understand and help improve those lives?
Well, again, I can't necessarily predict the future. I think we heard that early on, that I don't know what's going to happen in 10 years, but I certainly hope that we will know a lot more about the fundamental mechanisms of telomere biology, because one of the things that I'm excited about is that this really opens a new door to understand how the equilibrium is maintained. And if we don't understand how something works at a fundamental level, we can't have inroads into diagnostics and treatments. And so I'm hoping that in 10 years' time, we will know a lot more about the fundamental mechanisms so we can move forward toward those other treatments.
We see that every day here, people doing fundamental research upon which is built translational research and therefore hopefully diagnostics in the future. And Daniel, that is something that you've been giving us today, is an amazing glimpse into the world of RNA for liquid biopsy. So I guess my question for you is, why RNA for liquid biopsy? What potential advantages does it confer over the work that's been done so far, which is mainly focused around DNA and often methylation?
Yeah, I think for early detection, the biggest challenge is having a highly sensitive assay where when there's not a lot of signal, for example, in the blood, how can we really leverage all of the signal that's coming, especially from, for example, the transcriptome, where, as I mentioned in my talk, for a lot of the DNA, it's going to be degraded as soon as the cancer cell, for example, dies and releases its DNA into the circulation. And so you only have these very short 150 base pair fragments of DNA that actually survive. And so if you look at the RNA again, the advantage is that it's protected from degradation. And then for a lot of these RNAs, they're novel and specific to a disease like cancer. And so we can have highly specific signals that we can then leverage for our machine learning models that can give us better sensitivity and specificity for being able to diagnose cancer.
Fantastic. And I think there was a question from the audience about RNA. We all know that RNA has a bit of a reputation to be fragile, difficult to deal with. Does that hold you back from your science? Or how are you thinking about that in terms of the development of actual methods that might be distributed to more people in more places?
Yeah, so the RNA definitely is intact and full length, and we could only really determine that using, for example, nanopore sequencing, because if you do short read sequencing, you don't really get that true length of the molecule. And so when we did this study, we were quite surprised to see that there are many cell-free RNAs that are actually intact and protected from degradation. And so we think that there's going to be a lot of advantages for looking at different isoforms that are disease-specific, so we could better understand what's happening in these patients. As we're looking at the whole transcriptome, we know all the protein coding genes and pathways that are potentially dysregulated in these individuals. And so the hope is that not only would we get really accurate and highly sensitive detection of the disease, but also we would have information about how should we treat these patients, for example, that have mutations in KRAS or other oncogenes where there are potential therapeutic interventions that are more targeted. And so we think that RNA provides all of this information where it's not just diagnosis, but it's also really figuring out what are the right ways to treat patients. And then even once they're on therapy, these cancers will evolve. Patients will gain resistance to a lot of these monotherapies. And so even understanding mechanisms of resistance in vivo, we're able to see what are the RNAs that are turned on that potentially confer this resistance to, for example, a therapeutic. And so we think that there's a tremendous potential for diagnosis, for treatment, for monitoring cancer evolution. And as I mentioned, we are also starting to look at Alzheimer's disease, where we can not only hopefully detect it at an earlier stage, but just really understand what's happening in the brains of these patients, and could this inform novel therapeutic strategies as well.
I can see you nodding, Carol. I mean, it's amazing to have three scientists here. We have a conservation expert, we have a telomere expert, we have an RNA liquid biopsy expert. What are the areas of science that you have in common? Why is it important to bring people from all these different disciplines together here today? What do you benefit from talking to each other?
Well, nanopore analysis happens so fast, the change in what's happening with nanopore sequencing. So I'm really looking forward to talking to a lot of colleagues here and brainstorming like where things are going and what the next thing will be that we can take advantage of for our analysis.
Absolutely.
I was just thinking that, when you ask that question of like, where will something be in 10 years, right? Like you ask it to people that do human science, and it is hard to predict. It feels also new because you are the very cutting edge of everything. But when you think about plant and animal research, we're behind, right? Because we can almost see the future because we can see what's being done when you have an incredible telomere to telomere genome available. But now what if we could do that for these other species? So now you can kind of follow along and it's, I think it's exciting to be able to say, well, can we try that with this stuff? What would it reveal? Yeah.
Are you doing telomere to telomere approaches for your endangered species?
Yeah, I mean that would be the dream, right? To be able to get to that point. And so we're really excited about trying to take these protocols and see, what happens when you can't. Simple things. You don't have a centrifuge that can stay cold. Can you still do it? And so we play around a lot more with protocols that way to try to find alternatives to get things through. But I think that's the only limit, isn't there? Like, why not?
I can see a collaboration fostering right here on this sofa. It's so lovely to see. Well, I'm so sorry that's all we've got time for right now, but thank you very much. I know there'll be a lot of people out there having their lunch trying to grab you for a word and answer all the unanswered questions that you had at the end of your presentations. Thank you so much for joining us today.